engineers have exploited the tenacious nature of life by persuading it to build with silicon, the stuff of microchips. In a breakthrough study, researchers have tweaked a bacterial protein to knit together carbon and silicon, producing the basis for compounds used in everything from drugs to TV screens. The team claims that their enzyme is far more efficient than man-made catalysts and could reduce the cost of making the compounds and avoid using toxic material. By mutating bacterial DNA, the team at Caltech in San Diego has produced an enzyme which combines silicon and carbon. This process is key to making artificial compounds used in a range of industries, from pharmaceuticals to agriculture. As silicon-carbon bonds are not known to occur naturally, they are made in the lab by chemists. But in a first, the Caltech team has been able to tinker with proteins to do the job for them. We decided to get nature to do what only chemists could do, only better, said Frances Arnold, a researcher at Caltech and principal investigator on the study. The team isolated a protein found in a bacterium which bathes in the hot springs of Iceland, called cytochrome C. It is used by cells to share out electrons, but at low levels it was also seen to combine silicon and carbon. By tweaking the bacterium's genetic instructions for the protein, they were able to target an iron-containing region of the protein responsible for the silicon-carbon bonds. The resulting protein is an enzyme able to make bonds 15 times more efficiently than the best artificial catalyst produced by chemists. The same method has been used to produce enzymes for detergents and other common products. The team explains that the silicon enzyme could find a number of industrial applications and would avoid the use of precious metals and toxic solvents used in synthetic methods. By introducing the DNA for the enzyme to E. coli, they could have biofactories knitting together silicon and carbon for industrial compounds. This iron-based, genetically encoded catalyst is non-toxic, cheaper, and easier to modify compared to other catalysts used in chemical synthesis, explained Jennifer Can, a postdoctoral researcher at Caltech. The new reaction can also be done at room temperature and in water, she adds. Can is the lead author of the study, published this week in the journal Science. Although the breakthrough was reported at a conference earlier this year, life on Earth is centered on carbon, with the element forming the backbone of organic molecules in sugars, fats, and zoof other organic molecules. But scientists and science fiction writers have long pondered whether silicon could form the basis of life beyond our planet. The element sits just below our beloved carbon in the periodic table and shares similar characteristics. The researchers add that life could have evolved to use silicon by itself. This study shows how quickly nature can adapt to new challenges, said Professor Arnold. The DNA-encoded catalytic machinery of the cell can rapidly learn to promote new chemical reactions when we provide new reagents and the appropriate incentive in the form of artificial selection. Nature could have done this herself if she cared to.